Alright guys, so today we're going to be talking about DNA and RNA and what is the difference between the two. So here we got a nice picture over here of DNA, and you can see that DNA has been used to make RNA, and then you got RNA over here, and that's been used to use protein. But really kind of what's the difference between these two? I mean, we got some of these things, but what are some of the key differences? Alright, so I made this handy dandy little chart here to help us keep them straight. So DNA is double-stranded. Think D for double. Unfortunately, I don't have a handy-dandy word for RNA that starts with R. That means single-stranded, but RNA is single-stranded. All right, DNA. The sugar used in DNA is deoxyribose, right? And that's actually how DNA got its name, because DNA stands for deoxyribonucleic acid. And so that D in DNA literally stands for deoxyribose. RNA uses a different sugar, and the sugar is called ribose. And that is how RNA got its name. R stands for ribonucleic acid, right? Ribo is short for ribose. It's telling the type of sugar that is being used in RNA. Okay, next is your DNA mainly spends time in the nucleus. So going back to our picture over here, you can see, look, here's the nuclear membrane, and look, here's our DNA, it's in the nucleus. Now, RNA is made in the nucleus, but it doesn't stay there. It quickly moves from the nucleus outside of the nucleus into the cytoplasm, the part of the cell between the nuclear membrane and the cell membrane. So once again, RNA is in the cytoplasm, that's cytosol. DNA is long-lasting. Why? That is the instruction book on how to make every single protein in our body. And so we have to keep this for the life of the cell. Otherwise, we're not going to know how to make whatever it is that we need to do. RNA is going to be a different animal. RNA is going to be short-lasting. Why? So what happens is I take the DNA, I make a photocopy of it, and so now for the next 30 minutes or however long, I'm making that protein. And once I make enough of that protein, I get rid of that RNA. Why? I don't want to make any more of that protein. All right. Our DNA uses the following bases: adenine, thymine, cytosine, and guanine. RNA uses a lot of the same bases: adenine, cytosine, and guanine. The one base it uses difference is this U right here, uracil. So why does RNA use uracil and DNA use thymine? As far as I could tell, it's actually easier in terms of energy-wise for the cell to use uracil. It doesn't cost as much money or energy for the cell to use uracil. The problem with that is it's not going to be as durable. And so that's why we use uracil and RNA, is because it's cheaper and not as durable. But that doesn't matter because RNA is not going to last that long. Whereas DNA, it has to be durable, right? It has to pretty much last the life of the cell. And so we don't want to save energy by making a cheap base, because guess what? You're just going to have to keep fixing it over and over and over again as it breaks down. Okay, now one of the last major differences is that DNA comes from DNA, right? So when we need more DNA, we replicate it. When we need more RNA, right, we actually use DNA to make RNA. Now, you get weirdo viruses that will actually make RNA from RNA, but a normal human cell, RNA comes from DNA. All right, well, I hope that helps.